Hello, everyone, and welcome to our very first WooU event of 2023. And we are kicking off the year right with an incredibly innovative topic, which is antibody-drug conjugates for cancer. My name is Dr. Stephanie Wu. I will be your host for the evening, and I am the founder of Wu University. And it's now my pleasure to introduce our speaker for the evening. And this is really exciting because we've had, over 100 events over the last couple years. And this is our very first oncologist. And I'm really lucky because Dr. Borghaei responded to a tweet that I had sent him when I saw that he was speaking on this exact topic. So Dr. Borghaei, he earned his degree at the Philadelphia College of Osteopathic Medicine and completed a residency at Graduate Hospital in Philadelphia. And then since completing his fellowship training at Fox Chase, he's been involved in a number of clinical trials aimed at developing new antibody-based therapies and immunotherapies for patients with lung cancer. You can see how many accolades he has. And it's funny because earlier today I was talking to someone else, not even in the oncology industry, Philip from a different company, and he said that he knew Dr. Borghaei and how amazing of a speaker he is, an incredible doctor. So we're just so thrilled to have our very first oncologist present to WooU. So thank you so much, Dr. Borghaei, and it's lovely to have you.
Well, thank you so much, Dr. Wu, for the invitation. So as you heard, I am a medical oncologist. I concentrate on the treatment of patients with various lung cancers, and antibody-drug conjugates are one of the treatments that we have become very, very interested in. And I'll explain some of the enthusiasm that we have around this. These are my conflicts of interest as part of this CME program. You already saw a similar list and the mitigation strategy that was used. So why talk about lung cancer? So things have changed a lot in the world of lung cancer. About 20 years ago, when I was first a fellow at Fox Chase, the median survival for this disease was about a year. Now we are at a place where we're talking about a 32% 5-year survival for some of our patients who are getting treated with immunotherapies. We also recognize that lung cancer is not one disease. The pie chart and the table that you see here shows you the complexity and the heterogeneity of this disease. Non-small cell lung cancer now is divided not only by histology, but also by the molecular subtype. These driver mutations, as we call them, such as EGFR, ALK, and ROS1, as you see again on the table, have been identified in a number of different patients all over the world. And the interesting thing about these targeted therapies are that patients with advanced metastatic disease, even with brain metastases, can now be simply treated with oral agents at home. They don't have to come in. They don't have to be hooked up to IVs. So this is a major advancement that we've been able to bring to our patients in the past few years. And it literally is really in the past 10, 15 years that we've made all of this progress. And this is just the targeted therapies. And for patients who do not have an identifiable target, a molecularly driven target, we have immunotherapy with or without chemotherapy. So the advances have been significant, but unfortunately, with every drug comes additional toxicities. There's always room for improvement from a quality of life point of view, from survival point of view, response, and others. So we are actively looking for the newer and better treatments for our patients. That's why having newer agents becomes so important for us. We cannot say that we're curing everyone. The goal is to get to that cure, even for lung cancer, but we're not there, so we need new drugs. What you see here is simply a structure. For those of you who are not familiar with these, on the left-hand side, and I'm hoping that you can see my cursor, you see the structure of an antibody. Antibody is usually shown or depicted with a Y structure. There are two heads on top. These are the antigen binding portions of the antibody. So you identify a tumor antigen and you design an antibody to attach the particular antigen. And there is a tail, so to speak, we call the FC portion of the antibody. And this is where you get engagement of the immune effector cells, natural killer cells, the lymphocytes. The idea here is that this antibody can bind a specific tumor antigen. Older generation of antibodies, or sometimes newer ones, are designed to actually inhibit signaling through these particular antigens, and then bring the immune system into proximity of the specific tumor that's expressing your antigen of interest. What we've learned is that when this binding happens, some of the antibodies can actually enter the cell. So someone came up with a brilliant idea of attaching what we call, a warhead or a cytotoxic payload, and attach it to the antibody, such that when the antibody attacks or attaches to the antigen and is internalized, the drugs that are attached to the antibody are delivered to the cancer cells. So it's basically a method of delivering chemotherapy directly to the cancer cells that are expressing the antigen of interest. So the concept is rather simple, but we've had to learn a lot of different chemistry and biology to be able to come up with really good drugs. The antibody-drug conjugates have several components. Again, I'm going to go over this several times in a couple of different slides. So there's the antibody, there's the toxic payload, and there's a linker that actually attaches the toxic payload to the actual antibody. And this linker actually plays a very significant role in the stability of this compound, such that if this linker is not strong enough and it breaks and the drug is released into the systemic circulation, there could be problems. So the antibody brings us selectivity because you're attacking a specific tumor antigen, not all the cells. So hopefully the toxicity would be somewhat more limited because you're going after specific cancer cells. There's the linker that has the payload on it, and that provides the cytotoxicity that one requires to kill cancer cells. These are the sort of names of a whole bunch of different antibody-drug conjugates that are either in clinical development or some of which have been approved. Now, the slide says non-small cell lung cancer and small cell on the right-hand side, both diseases for which we really need better drugs. But I have to point out that some of these targets, such as HER2 and HER3, are very relevant in other diseases. For instance, for those of you who might have some familiarity with the cancer literature, HER2 is a favorite target for breast cancer because many breast cancers express this. HER3, the same. Many of these others, such as TROP2 or CEACAM, I'm going to talk about these antibodies, can be expressed by a variety of different malignancies, and therefore the relevance could be to a number of different solid tumors. In the world of solid tumors, we have a couple of antibody-drug conjugates approved, one specifically in breast cancer, and that's this ADO, trastuzumab, emtansine, or DM1. And then we have trastuzumab deruxtecan. That's the newest antibody-drug conjugate. This one is the only one approved in lung cancer right now. We're going to see some of the data for that, but it also has pretty significant activity in breast cancer. We also have sacituzumab govitecan, which is a TROP2 antibody-drug conjugate. It's also approved in other cancers and under investigation in the world of lung cancer. This slide then shows the binding of the antibody to the specific tumor antigens. Some of these tumor antigens have, in fact, biological relevance, meaning that if you have an antibody that can block signaling, you can stop a cell from growing by simply attaching the antibody to it. Some do not have as much of a signaling moiety, but because they're expressed on the cell surface of many different malignancies, they provide a good target for the antibody-drug conjugates, allowing us to get cytotoxic drug more specifically into the tumor cell. So here the binding happens, and the antibody comes in contact with the tumor antigen. The ADC is then internalized, comes into the cell. And then once inside the cell, there are a whole bunch of different enzymes that act on the antibody-drug conjugate, releasing the toxin and therefore killing the cancer cell. There's a little bit of a bystander effect that could happen. And what we mean by that is the toxic moiety can actually permeate from out of this particular cell that's dead and enter the neighboring cells. So not every cell, cancer cell in a tumor, has to be bound by the antibody. So there is a little bit of a bystander effect that, this slide perhaps shows a little bit better. So this is a multi-step process. It's easily seen here. We can identify at least 6 different levels, let's say, that the ADC has to go through to be effective. What it also means is that there are 6 separate ways where the cancer cell can interfere, or the body can interfere with the functioning of antibody-drug conjugates. Therefore, resistance to these drugs is also unfortunately common. Moreover, the toxicities seem to be associated with the toxic moiety that's actually used, such that, as you can see, if some of these toxin actually gets into the circulation, it can cause real side effects. Now, for the most part, these toxins are actually so strong and so potent that we never use them by themselves as chemotherapies, because they're very, very toxic. We use very small amounts of them here in the ADCs. So some of the advancements that have been made recently is to come up with better toxins, and again, better linkers that keep these things together. So why is this concept of here, the killing the neighboring cell is important? Well, most solid tumors are very heterogeneous. So what you see on the left-hand side on the control is a mixture of a couple of different breast cancers. Some express HER2, shown with the brown staining. This is an immunohistochemistry. And some that are HER2 negative. So that's the sort of the light blue cells. And this is obviously an experiment done to show the selectivity or the efficacy of these drugs. So some HER2 positive, some HER2 negative cell lines. If you use this particular ADC that targets HER2 positive cells, you very effectively get rid of all the brown staining. So all the HER2 positive cells are gone, but you're left with the HER2 negative cells, because this particular one doesn't have as much of a bystander effect. On the other hand, if you use this other antibody, which is the trastuzumab, the deruxtecan, you see that both HER2 positive and HER2 negative cells can actually be eliminated by using an antibody-drug conjugate like this. Tumor cells, solid tumor in particular, are extremely heterogeneous. You never get uniform staining of specific IHCs, and there is a variability, very strong IHC positivity to somewhat positivity, and really every drug has to be developed along the pathway of these biomarker development. And again, I'm going to show you a couple of ADCs where, strong presentation by the tumor of the antigen could actually be important. So this sort of establishes the fact that if you have some bystander effect from a cancer killing point of view, this is a better strategy, and you might want it better. The other thing that we have to keep in mind is the number of toxins that we can attach to these particular antibodies. Why is that important? Well, as I said, we use minute amount of these toxins. So if you have 1 or 2 toxins attached to the antibody, the clinical activity might be lower. You can actually come up with the chemistry, which is kind of actually tricky, and it took us a while to figure that out. And by us, obviously, I mean people who do antibody engineering and all that. And for a portion of my career, I actually did antibody development, and I did make my own antibody and my own ADCs and all that. And it really is labor intensive, obviously, in an academic lab, not in an industry. But you can get more drugs on the antibody that actually could increase the efficacy, because again, you want to get as much toxin into the cancer cells to sort of kill them. So the first generations of ADCs came to the clinic about 10, 15 years ago. We liked them. They were not quite as active as we wanted. There were a lot of toxicities. The ADCs went out of favor for a while, and now they have made a significant comeback. There are many ADCs in clinical development, various studies, better antibody targeting, better linker, more drugs attached to the antibodies, and also somewhat better drugs. And again, the linker chemistry that are not allowing the toxin to be released sort of willy-nilly into the circulation is very, very helpful. On the right-hand side, you see that we're coming up with what we call bispecific ADCs. You can bind two separate tumor antigens and actually maybe a tumor antigen and some immune cells. Why is that better? Well, because it increases your specificity when you have two different antigens that you can select, and therefore, again, hopefully a better killing activity against the cancer cells. We could have a couple of different payloads attached, perhaps one that immune stimulating, one that can be directly cytotoxic, and also going back to an old favorite, and that's attaching radionuclides to an antibody and using this radioactive particles to, again, directly kill cancer cells and some of the neighboring cells. So a whole bunch of different structures are upcoming. This is just a timeline of where we've been and what we come, again, to indicate that, it took quite a while to get to this first stage in 1990s. And then all of a sudden, in the mid 2010, 2015, we started seeing a whole bunch of antibodies coming out with ADCs, I should say. And then there's been an explosion of a whole number of these drugs. So some of the favorite targets that, again, can be relevant in lung cancer and other diseases are shown here. But again, this is just an example. In the interest of time and all that, I'm not going to have an exhaustive list. But HER2, we talked about, relevant in lung cancer, relevant in breast cancer, HER3, a number of different tumors, TROP2, large number of cancer cells can be producing CCAM. This is, again, something that can be expressed on colon cancer, lung cancer. c-MET. MET is one of the major antigens and targets that we're interested in. We have oral agents, we have antibody-drug conjugates, we have bispecific antibodies. Again, a lot of interest in MET in particular. And this is just a table of comparing a number of these in terms of what the name of the ADC is, what is the antibody component of this. The linkers come in a couple of varieties, what we call them cleavable and non-cleavable. Most of these, as you see, are cleavable. And the toxic payload is a little bit different. The roxacan and SN38 have been used now more extensively. So some of the newer antibodies of this, basically, these are, let's call them cell cycle inhibitors, and they interfere with the cell division and the whole machinery. And then DAR is a drug to antibody ratio. As you see, we can be as low as 3.1 drugs per antibody, as high as 8. So again, in some of the literature that you encounter with this drug, with this sort of class of drugs, having more toxin, if possible, can be better. Toxins can be bulky. So sometimes if you select a kind of toxin that's really bulky from a chemical structure point of view, you just can't attach too much, and they just mesh together and distort the whole structure there. So it's a little bit problematic. But if it is not as bulky, then it is possible to get some on it. And again, the chemistry here has to get worked out quite extensively. Brentuximab vedotin is an ADC that's been around for use in hematologic malignancies. It's an approved agent, as you can see here, for patients with relapse refractory Hodgkin's lymphoma. It targets an antigen called CD30. In the world of hematologic malignancies, leukemias, lymphomas, and things like that, we have a little bit less tumor heterogeneity. There's more of a uniform expression of the specific antigen. And therefore, these antibody-drug conjugates can be quite useful in management of patients with various hematologic malignancies. And obviously, this is one. And as I said, it's been around, and still some investigations are ongoing to sort of extend the role of this antibody in some of the other heme malignancies that express CD30. And from a theoretical point of view, if you can find a cell surface antigen that's specifically expressed on a cancer cell and doesn't have any expression in normal cells and normal tissue, and there are quite a few of these, then we should be able to have an ADC or at least an antibody. The key thing is that the antigen has to be internalized upon binding, and that internalization process is not something that happens uniformly for all cancer antigens. So again, makes it a little bit more difficult to select the right target for the antibody to get that internalization. And again, as I was pointed out before, there are a couple of these ADCs approved in solid tumors, mostly in breast cancer, and very recently one in the world of lung cancer. So what are some of the toxicities? Well, because of the cytotoxic component, which is basically chemotherapy. So think of ADCs as a targeted delivery of standard chemotherapy drugs. The side effect profile is by and large very similar across these ADCs, include hematologic toxicities, so thrombocytopenia, leukopenia, GI, so there's a lot of nausea and vomiting, not a lot, but associated anyway with these drugs. Again, very similar to chemo. Some can actually cause alopecia, again, very similar to cytotoxics. But what might be of interest to this particular group is also the fact that we do get ocular toxicities in terms of keratopathy in a number of patients here. So these are the sort of a large subgroup of toxicities reported with these agents. And again, I have multiple slides going through this just to show you that some of these tumor antigens are actually a bit more complicated because they can form dimers, either homodimers or heterodimers with different members of the family. Again, HER2 is a classic example of all of that. For some of these, we have other active oral agents, but obviously the oral agents, the tyrosine kinases, act on a different component of the whole signaling pathway. But the antibody-drug conjugates are usually attaching themselves to the extracellular domain, where on the surface of the cancer cells, you have the expression of these particular tumor antigens. So this particular ADC, let's just call it TDM1, is an ADC that was used in the world of breast cancer, approved, and then it was tested in lung cancer because there are a portion of lung cancers that actually have HER2 either mutation, which is what the subject and the title is here. That is about 2 to 3%, maybe 4% of adenocarcinomas in the world of lung cancer. And then there are some lung cancers that actually have overexpression of HER2. So overexpression and mutation are a little bit of a different concept. The actual HER2 mutation suggests that we did the sequencing and we actually found base changes within the DNA there. So in the HER2 gene, so it's a mutated HER2. This particular ADC had some activity. So again, for those of you who might not be familiar, the study is just sort of explained. We had HER2 mutation and HER2 amplified subgroup of patients. The dosing was determined based on some of the prior studies. What we have here in the middle, again, hoping that you can see my cursor, we call a waterfall plot, and just because it looks like there's waterfall going down. The dotted line saying 30%, that's 30% reduction in the size of the actual tumors that you're measuring. By convention, we call that a partial response if a tumor is reduced by about 30%. Obviously, a complete response would be 100% reduction. You can't see anything. So this is waterfall. 0 is the baseline. Obviously, everything that goes above suggests that patients have progressive disease. Yellow in this is stable disease, so not reaching partial response, but not progressing. Obviously here, blue is called a partial response. And then what we have here is called a swimmer's plot. It's because it looks like the swimming lane. And this gives you a little bit of an idea of the durability. So on the bottom, we have the time scale in terms of, how many weeks or days or months, depending on the drug, the activity lasted. And usually when we have these red dots or other triangles or something like that here, as you see on the swimmer's plot, that indicates when the actual responses were first seen in these patients. So for the majority of these patients, responses can happen relatively early, but then there's some that have responses a little bit later. So they might have a stable disease and all of a sudden, after several cycles, they have stable disease. So waterfall plot and swimmer plot, and then you're going to see something we call a spider plot. I was kind of hoping after swimmer plot, we have the whole Olympic sports plots, but that hasn't panned out. I have to work on that, wrestlers or soccer plot and things like that. And this is your standard Kaplan-Meier curve for the PFS, basically showing the progression-free survival of these patients. So by and large, I would say when I see something like this, I would say, it's not bad, but it's not like this is, burning the house down. It does seem to have some activity. Some of the activities are sort of lasting, which is good. We want to see at least stability and some sort of a partial response. But the median PFS and all that doesn't seem to be all that great. So if you look at the 50% line, you come here is around 4.5, 5 months, which for a study like this, where we take patients who've gone through multiple prior lines of therapy, and therefore they have a very resistant tumor left over. So again, think about it. These patients have had frontline therapy, they've had second line therapy, and then they're usually candidates for these experimental protocols. So some of these patients might have had 3 or 4 prior lines of therapy, and then the cancer that's left is actually a very resistant cancer. So having something that gives you a 20-30% response rate that lasts, for several months, it's a reasonable level of activity. We're not happy with that, so other antibody-drug conjugates were developed. This is trastuzumab deruxtecan. This one uses a different toxic payload, which is the deruxtecan moiety. It has more antibody-drug ratio, so about 8 drugs are attached to the antibody, but it still targets the same HER2. So this went through several different clinical trials in a number of malignancies, including lung, and this is just a sort of the difference between the TDM1, which is the antibody we just looked at, the ADC we just looked at, and DXD, which is the ADC we're talking about right now. Well, in a very well-conducted phase 2 study, about 52 patients with non-small cell lung cancer with HER2 mutation, response rate for this ADC, as you can see here, is around 54 to 58%, with a median duration response of around 8.7 months, which is pretty meaningful when it comes to HER2 mutation. Again, rare patient population by standards of lung cancer. Again, it's only adenocarcinomas, and under that category, 2 to 4% of patients would have a HER2 mutation. But since we do molecular testing on every lung cancer patients at this one, and this is an exhaustive 300 gene panel, usually looking for these targets that I showed you in the beginning, we can find these alterations even when they're rare, because everybody is getting a molecular testing panel of genomic assays. Therefore, this is, a reasonable drug and with good clinical activity. Again, the waterfall plots just showing you a couple of different doses were tested, and the dose that has good activity with less toxicity, in this case, the 5.4 milligram per kilogram, is usually the dose that goes through. Again, same waterfall plot. Notice there are very few people on top of this 0, meaning very few people had progression, at least on this study. Again, we consider this to be good. And in lung cancer, we hardly ever get a complete response. It's very unusual. You see it from time to time with targeted therapies. Here, we didn't really hit necessarily CR, maybe 1 or 2 patients, but nonetheless, these are pretty deep responses. So what about toxicity? Well, as you see here with the lower dose, there's a little bit less toxicity. So these sort of what we call tornado plots give you a side-by-side comparison of a couple of different drugs, or in this case, a couple of different doses. So there's definitely less toxicity and good clinical activity with a lower dose of the drug, and that's what's chosen to be taken forward. From a clinical setting, sort of evaluating these drugs, you look at the discontinuation rate, how many patients actually stop it just because the drug was too toxic, and that's around 8% at this dose. How many of these patients needed a dose reduction? About 10%. How many was actually the whole drug interruption? That's about 14%, and these numbers are fairly reasonable for, again, the second, third line setting. This drug comes with a special toxicity of causing interstitial lung disease, or ILD, which is a non-specific term sort of suggesting an inflammatory process that, again, is reported with many, many drugs that are used in the world of lung cancer, even some old-fashioned chemotherapy drugs. So if you check the literature, almost everything we use in the world of lung cancer can be associated with this inflammatory pneumonitis, and many different reasons why that could be. This particular drug does have ILD associated with it. And for that reason, patients have to be monitored very, very closely, because this can progress rather rapidly and it could cause major issues for patients. And again, for those of you who might not be familiar, grade 1 is minimal symptoms. Grade 5 is unfortunately a fatal event that sometimes is reported on these clinical trials. Grade 4 is something that's so serious that patient usually requires hospitalizations. Grade 3, again, serious in need of treatment, maybe not necessarily leading to hospitalization, but it could happen. Grades 1 and 2 are the ones that we consider to be mostly manageable from an outpatient point of view. Nonetheless, I would highlight the chronic grade 2 toxicities, especially if it hits the eye, and all of that could be extremely annoying, could interfere with patients' quality of life. And therefore, it's something that it's not very desirable. Grade 1, 2 toxicities can be overcome and are self-limiting. Obviously, would be better for a patient's quality of life. But unfortunately, again, all of these drugs have some degree of toxicity associated with them. So this drug was approved by the FDA August of 2022. And again, just so that you know, some of your patients that you see could be exposed to this. Same antibody drug conjugate, this time targeting something called a HER3, which is another antigen in the, or another receptor in the HER family, which is one of the best studied pathways and families in lung cancer. This one is also very interesting. I know this is a really busy slide, and I'm sorry, I just couldn't make this simpler. But what I want to show you is that, again, this is lung cancer. This is patients with a lot of disease that we, as a term, we call it refractory disease, meaning multiple lines of therapy and the cancer keeps coming back. And again, every time the cancer comes back, could have new mutations and could be a little bit more resistant. So on the bottom here, these patients all had, with lung cancer, some degree of mutation in another member of the HER family called EGFR. These patients, for the most part, were treated with oral agents with good clinical activity, sometimes 2 years, 2.5 years on a simple pill with good activity, including CNS. And then as a result of these treatments, they acquire additional mutations, as you see on the bottom. There are amplifications, so some genes are more expressed and not necessarily mutated. And then some other pathways can become mutated. And this particular drug seems to have activity across a variety of these complex mutational landscapes for these patients. And this is actually, something we need. There's an unmet need for patients with these cancers after progression on oral tyrosine kinases. So having an option to go to once these medications have started working, or once standard chemotherapy, or once standard immunotherapy doesn't work, it's essential. Now, in this case, for patients with EGFR mutations, we don't use immunotherapy because immunotherapies don't work in these tumors. So again, highlights the fact that there are, hundreds of thousands of patients worldwide that need better treatment options, and these drugs can play a very valuable role. So 39% response rate with about 7 months duration of response, and a PFS, as you see here, is a pretty reasonable activity for a single drug. And obviously, the goal would be to improve on all of this. Toxicities here, again, you have to worry about the ILD. That's always on top of our list. But again, very much similar to standard chemo, drop in platelets, white counts, and all of that is all associated with this. I've mentioned TROP2 can be a relevant target because it has a signaling pathway, as you see here. Many of these tumor antigens can interact with a number of different signaling pathways that are known to be associated with tumor progression, cancer growth, invasion. You see that in some of the adenocarcinomas, expression or lack of expression of TROP2 can have a prognostic significance, and therefore this could be a reasonable target for antibody-drug conjugate. This drug, sacituzumab govitecan, has undergone evaluation. Again, this is the spider plot that I was telling you about. This is the standard waterfall plot that you're familiar with now, and this is our standard swimmer's plot. The toxic payload here is SN38, which actually is a metabolite of a topoisomerase 1 inhibitor. There is a chemo drug called irinotecan, which when used systemically, it's broken down in the body, and SN38 is one of the byproducts. And again, the activity shown here in the table summarized for you, indicating that, again, this is a reasonable antibody-drug conjugate. Only 54 patients. Obviously, we need to expand on that. Response rate is 17%. It's okay, but I would have liked to see a higher response rate. Obviously, I think response still matters, particularly for patients who have symptoms from their disease, with an overall survival, at least based on the study, of around 7 months or so. Which again, it's an option, but I wouldn't jump up and down and saying this is the best drug I want to have right now. That's what I want to use. So then we have another generation of these ADCs coming. It is undergoing an extensive evaluation, multiple cohorts of patients at different dose levels, as you see on this. And here there's the sort of the summary of responses at various dose levels. We actually have to go through this dose evaluation kind of carefully because it doesn't necessarily mean that a higher dose is better. You don't necessarily get a significant bump in response going from 4 to 6 to 8. But I can tell you the toxicity definitely goes up when you have more of these antibodies, particularly when they have a lot of drugs attached to them. So you have to come up with a balance. Again, patient quality of life is really important. You don't want to give a drug, even if it's effective, if the patient is going to be in the hospital all the time with various toxicity. So finding the right dose takes a lot of trial. So here, evaluating this particular antibody, and I have highlighted here, one of the toxicities we begin to see is this dry eye component, which again, particularly for older patients, this becomes really problematic. Some of them, it's such a difficult thing that driving becomes a problem. So again, we have to pay attention to the toxicities that the patients are suffering from. Here, again, you see perhaps a little bit of a higher percentage of patients having nausea, for instance. So the percentages are shown down here. Stomatitis is mouth sores. Patients get alopecia. Again, very much similar to what you get for standard cytotoxic chemotherapy. These drugs have real adverse events, and we do have to pay attention to them. You look here, 98% of patients, or 100% of them, had some sort of adverse event. TEAA is treatment emergent adverse events. At least something was reported. So majority of these patients end up having toxicities. For many of these antibody-drug conjugate studies, almost all the ones that we have at Fox Chase, we actually partner with our ophthalmology group because we require frequent eye exams before the treatment as our baseline. And then depending on the ADC at various time points throughout the study, and sometimes for several months after the patient has discontinued the treatment, we require a pretty extensive eye exam just to make sure we haven't done more damage. And this is like one of those areas where, as an oncologist, I don't know how to do a proper eye exam. So this is where we partner again with our ophthalmology colleagues very, very heavily. And then given the popularity of these drugs, the demand to have an ophthalmologist available for evaluation has become even more. I know how to manage nausea. I know how to manage stomatitis, but as an oncologist, I'm never going to be able to sort of do the kind of eye exam that you can do for us. So having that collaboration and close work with the ophthalmology group has been very, very essential for the development of these drugs for all of us. And again, from personal experience, it's been great to be able to work with the ophthalmology groups close to us. The next ADC is the CCAM. Why am I interested in this? Well, again, this is a relevant target. It's expressed again in a variety of different cancers. Again, the linker here is very specific. And I'm not mentioning any company names and all that because it's really irrelevant, but every company has its own version of an antibody, their own version of a linker, some better, some not. And you really sometimes don't know until the actual studies have done. And then the toxic moiety here is this DM4, which is a different toxin. This is a maytansinoid derivative that basically inhibits polymerization of tubulins and therefore causes a cell cycle arrest. And the immunohistochemistry of a variety of cancers, including stomach and pancreas and cervix and lung cancer, indicating that the target is expressed in a variety of different tumors. And then this also has gone through a standard phase 1-2 evaluation. So we start at low doses, gradually build up to something that we think is a reasonable dose, and then expand specific cohorts of patients. Here, we're trying to develop a biomarker as opposed to some of the other drugs. Meaning that we're doing IHCs on patients, identifying those that have high expression of the particular target or moderate expression, and sort of dividing them into these cohorts and evaluating the activity of the drug in the biomarker selected patient population, the high versus low. And again, I'm not going to go into the details of the high and low. These are, again, things that we sort of decide as we're designing the studies and how we want to interpret it. It can vary from company to company. So here, if you look at the high expressing tumors in tau versus the moderate expressing, there's clearly a difference, right? 20% versus 7% or so. So This does tell me that this ADC should perhaps be targeted to patients that have a high expression of the tumor antigen, and maybe the moderate would not benefit quite as much from this. And again, this has to be built on and has to be evaluated further. The median duration of response is a little bit different, right? You have moderate and high expressings, and you can see the differences in the median duration. So again, perhaps tumor selection would be important. Now here, we begin to see a little bit more sort of ophthalmologic toxicities, and this is sort of highlighted here. So first of all, almost everybody gets some level of toxicity, unfortunately, from these drugs. But if you concentrate on corneal adverse events, and these are, again, reported by our ophthalmologist colleagues, you get keratitis, keratopathy, up to 38% of patients, and of those, 11% are grade 3 or higher. So majority are still grades 1 and 2, maybe can be managed with holding a dose or something to that effect. But 11% grade 3 or higher keratitis is pretty serious. These patients have to be seen and followed quite regularly to make sure these toxicities actually resolve. So it's a problem for us. Neuropathy can be seen, diarrhea. Again, some of these are very similar to the other cytotoxics. The same thing with some of the myelosuppressive effects that you see from these drugs. And this is a picture that was provided by some of our colleagues from Spain as to what this DM4, which is a toxin here, induced eye damage could be. This is described as a microcytic corneal dystrophy. I'm not even going to attempt to explain this because I'm sure you guys can give me a 3-hour lecture on what this is. But these are the kind of pictures that we see. So these ocular events are summarized in this table here for you, right? These are specific to this particular ADC and this particular toxin, right? These are reversible. First occurrence can be during the first phases of the treatment, the first 4 cycles or so. And then, the troubling thing for me is that at least now it looks like primary prophylaxis is not effective. So there's nothing really you can do to prevent this from happening, at least based on the experience that we have so far with these drugs. So this is sort of hopefully provides a little an example of what could be happening. Now, either you're participating in academic centers where they're doing these studies and you're going to see these patients when they're on study, or 5 years down the road, if this drug gets approved, this could be walking into your office one day and you could get a call from your friendly neighborhood oncologist saying, hey, I'm using this drug and something happening. My hope is that if these drugs become approved, we actually going to have an algorithm say, nobody should get these drugs until they've had an established relationship with an ophthalmologist, go through the evaluation, just be seen and evaluated on a regular basis. So that has to get worked out. But this is what we're facing with right now. Advancing. Again, just providing you a little bit more example of, how these keratopathy and keratitis and all that, or these punctate keratitis, which again, I'm not as familiar with it as you are, obviously. These are defined toxicities so that if at a certain dose, like here at 120 milligram dose, 3 out of 8 patients have this toxicity. That's a no-go. That's a toxic dose, and we have to go back down a dose, which is 100 milligram. But even at 100 milligram, you could have some toxicities. Although the first 6 patients here didn't have any, so this sort of is selected as a dose that we might be able to take forward. But the dosing here could be an important indicator or predictor of what kind of toxicities. And this is, again, the rest of some of these other toxicities that have been reported, just to show you an example of it. Now, we decided to say, what if we do a loading dose followed by a lower dose of the drug? And why the loading dose? Well, we're trying to saturate all the antigens that are expressed on all of the tumor cells. So the idea here is that not to let any tumor cell escape. So if you have a big loading dose coming in and then smaller doses continuing, can we have better clinical activity? That's still a question, but that's what we're examining here. And then the loading dose, as you see here, could be anywhere from 120 to all the way to 170 milligram based on this clinical trial. And obviously at various doses, you still do get these keratopathy, keratitis, and dry eyes, in addition to some of the other toxicities. So these are color-coded based on the dosing that you see here. So at 170 milligram loading dose, which is this light green, we have 4 out of 5 patients having, significant eye toxicity. So these things take time and a lot of patient participation. These are brave patients who agreed to go on these clinical trials for us to be able to evaluate them with the hope of finding a drug that could be tolerable and active. So it really is a brave thing for patients to participate in these studies. Same thing here, again, indicating that the various loading doses and follow-ups are still associated with the toxicities, but you have to come up with the balance of what is acceptable toxicity without compromising the quality of life too much versus the clinical activity, and is it worth it? Many of these drugs don't make it, quite frankly, because they're just too toxic and not active enough. But the problem comes when you find something that really is active, and it could be a potential drug, but it has a lot of toxicities, and how do you mitigate that? And that's, again, what we go through with clinical trials and all that. So in summary, these are clinically useful drugs, definitely. In solid tumors, a lot of them are under investigation. We're going to see more of these approved, and I'm pretty sure of it. The toxicity is very much related to the payload that's been used, and I'm hoping that I have been able to convey that. And to tell you that, there are combination studies. So some of these could end up being practice-changing ADCs. We're not quite there yet, but some novel clinical trials are underway to evaluate all of these, and the hope is that at least some of these drugs can be that real drug that you're looking for with manageable toxicity. So thanks for your attention, and I hope you learned something from this, and I'm here for questions.
Excellent. That was such a great lecture to kick off the new year. We're always wanting to know more as eye doctors. We want to know kind of what's going on in medicine. And I remember when I first heard about antibody-drug conjugates, I was like, I've never even heard of this. I don't remember learning about it in school as far as different cancer treatments. Not that we learn a ton in school about that, but I had never heard of this before. And then kind of doing the research, it sounded so interesting that these are really unique, novel ways to treat cancer. It sounds like, I can't remember how many are FDA approved to treat cancer, but it sounds like there's like 100 of them that are like in the pipeline that are going for FDA approval in the next few years. So this could really be revolutionary as far as cancer treatment. And as eye doctors, we really need to be aware of these patients that might be on these treatments and kind of co-managing them. So I think you did a beautiful job explaining what ADCs are, kind of going through the mechanism of action, why they work, and just some of the different adverse events. And we do have some questions if you have a couple minutes.
Sure.
Okay, so I did not make this up. This is from Dr. Lung. I thought it was funny because you specialize in lung cancer. So Dr. Lung wants to know, what is a typical ADC treatment plan like? And typically, how long we will know if the treatment is effective or not?
So the drugs, because of the half-life of the antibody and the toxin and all that, are given, first of all, these are all IV medications, so you need a good IV access. If I understand the question correctly, they can be given anywhere from once every 3 weeks, so once every 4 weeks. There's one that we are actually administering once every 2 weeks. Just again, because of some of the pharmacodynamics of the drugs. On clinical trials, if it's a phase 1, we start at a low dose and patients are divided into these subgroups and we gradually go up. And approved drugs, if the question is, what is the course of treatment? That's somewhat open-ended. There are some studies that have a finite amount of treatment prescribed, meaning that you get the treatment for 2 years and then no more after that. In the world of metastatic solid tumor, especially lung cancer, for some of these drugs, we technically can continue the drug as long as the cancer is responding and as long as the patient has acceptable, tolerable toxicity. So if the toxicity is too much and it's just not safe to continue the treatment, we stop. And obviously, if the scans that we do, and we live and die by these CAT scans, show that the cancer is progressing and spreading and all that, then we stop the treatment.
That makes total sense. Okay, maybe you don't know this, but a couple doctors have asked the same question. Dr. Zerbanopoulos and Dr. Gento. The most common ocular malignancies that we see are melanoma. Do you know if there's any ADC meds that are in the pipeline for melanoma or ocular cancers?
I have not come across any specific ADCs for ocular melanoma. So that is something that I would say we would have seen it, because we do see a few patients like that, but I'm not aware of any. It doesn't mean that there's absolutely nothing out there, but I haven't come across them.
Excellent. Dr. Shovlin wants to know, beyond the cornea, we have seen 2 patients this past year with retinal vascular occlusion, one BRAF inhibitor, the other MEK inhibitor. Both, I think, were breast cancer. Have you had confirmation of any of your patients experiencing these relatively rare events with any of the other ADCs?
No, I haven't really seen the vascular occlusion, but the BRAF and MEK inhibitors are known to be associated with some of these toxicities. So that's a very specific targeted therapy type of adverse events that you are describing here. I've not seen anything like that in the ADC literature, but keep in mind that these are still relatively young in the world of solid tumors and lung cancer. So what I mean by that is sometimes with exposure of more patients to these drugs, you do find some of these more rare adverse events. But so far, based on, again, the studies that I've showed you, 50 patients there, 60 patients there, we haven't necessarily come across that. But it doesn't mean that's not going to happen.
Yeah. Yeah, like you said, it's just so new that who knows, there's just the sky's the limit.
Yeah.
Another great question from Dr. Shovlin, are you getting any resistance from coverage from major insurance payers for the ADCs that are already approved?
No, actually, we have not had any resistance. So all of the national guidelines endorse the use of any of the drugs that are FDA approved. So we haven't really had issues. Sometimes what we have issues with is high copays for some of the drugs, because as you know, some of the insurance companies still have a copay, not as much for cancer drugs that are IV administered in the cancer center, but we still have some issues with the copays and all that. And that, we have several different mechanisms to go through it. But once a drug is FDA approved, Medicare covers them, and all of the major carriers, can decline paying for an FDA approved regimen if applied appropriately.
That's great, because I know, we have so many treatments that we're always fighting for patients, and, it just gets denied. We were just talking about Oxervate last month, and, that's a very expensive treatment, and a lot of patients get denied by their insurance. It's so frustrating for us because these are things that the patients need. So that's excellent that it doesn't seem like you're having a huge amount of pushback. That's great.
Yeah, no, we're lucky in that regard. Again, I can't think of an FDA-approved drug that I've had to fight for necessarily to get it into a patient. Again, copays are different, but having access to the drug has not been quite as bad of an issue for us. We do fight with insurance companies, but like over how many CAT scans we can do, and can I do a brain MRI now? And if I want something stat, it has to be pre-approved. So it's a mess. But the drug has been relatively better, and these are very, very expensive drugs, I must add.
Of course. Yeah. Well, I know a lot of docs tonight are very jealous of hearing that.
Yeah.
Another question on how quickly do the toxicities show up after starting to take the ADCs?
Highly variable. Majority of the ocular toxicities were seen within the first 4 cycles of treatment. And if I remember correctly, for this particular drug, the cycles is every 3 weeks. So the first 12 weeks, you see a lot of these toxicities. That is hard to predict. Majority of adverse events that we see for many of the cancer drugs happen during the first few cycles of treatment, the first few months or so. It doesn't mean that it doesn't happen later on, but for some reason, you seem to get a little bit less of toxicity, especially when it comes to like immunotherapy drugs and all that. For some of the ADCs, my guess is that some of these toxicities could in fact be cumulative, such that if it doesn't happen with the first 2 or 3 treatments, as the patient stays on it longer, there could be a cumulative effect that could lead to some of the toxicities. But that's just my gut feeling about using these ADCs and all of that. But there really isn't necessarily a rhyme or reason as to when the toxicities can happen. We monitor these patients, as long as they're on these drugs, very, very carefully, because we know that adverse events can happen at any time.
Yep, that makes sense. Dr. Christensen wants to know, is there any data about the dry eye being induced by the drug or whatever ocular event? And then can that be reversed once the drug is stopped? Or does it usually get reversed, or does a patient usually continue to have problems long term?
Majority appear to be reversible. They require interventions, steroids and things of that effect. So the studies that I showed you, majority of these adverse events were reversible. We haven't had too many examples of permanent damage. Now, if you comb the literature, there are definitely patients that have long-lasting issues with some of the older ADCs and all that. With these newer ones, I haven't necessarily seen the data suggesting that in a large percentage of patients that the adverse events were not reversible. Again, I would caution saying the experience is very limited with these drugs. So it is unfortunately possible that we're going to get somebody with permanent damage.
Mm-hmm. Yep. Yeah, that makes sense. I mean, sometimes in clinic see once we stop a certain drug, their eyes will go back to normal. But then, of course, there's other cases where the damage is done and the meibomian glands or whatever thing is just not working. The structure is kind of damaged, permanently. So I think that's something good to know, and that gives us hope that maybe if they stop the drug, things will kind of go back to normal. Yeah. That's great.
Yeah.
Excellent. The other questions are on ocular events and kind of going into more specifics. So I'm going to keep those for next week because we've got an ophthalmologist that's going to be on, and we're going to be talking about ADCs and some of the ocular side effects and some of the treatment protocols. So we will save your guys' questions for next week.